Okay, so hopefully that's all coming up all good. So, hello, I am indeed Rowan Herridge, and I'm here from the University of Otago, and I'm going to talk to you about new breeding techniques, which are new ways to breed things. We're going to go through kind of slowly, get everyone up to speed. So the first topic that we'll cover is breeding in general. Potatoes do sometimes go through breeding processes, and people do make new varieties. So we'll start everyone on sort of the ground level and get breeding and kind of the idea of genetics as well incorporated there. And then we'll touch on a couple of different gene technologies, new breeding techniques being one of them. We'll then focus in on some new breeding techniques and how they work and what that really means. And finally, like, what is Otago doing? Why are we interested in this? And so the final little bit is about F1 hybrids and what we're doing at Otago University. So I show this this diagram here to my third-year students to hopefully teach them about how breeding works. So people breed lots of things. People breed plants and people breed animals. And the goal of this is really what's called genetic gain, which is gains in the properties or the traits of the plant or animals that are inherent to the plant or the animal, right, encoded in the DNA. There are lots of other ways you can make gains to your productivity by adding more fertilizer. So agronomic techniques also make gains. But what we're doing when we're breeding is we're trying to make plants that have better genetics than what you had before. And so the first part of any breeding strategy is to cross two things together. So cross a couple of different varieties, a couple of different potatoes, whatever, and get something new. So everything, all the DNA at this stage gets jumbled up and new stuff comes out. So that's the offspring. You then go through a series of analyses to try and figure out which of those new combinations, which of those new varieties is the good one. So you do field trials, you analyze the data and make sure that whatever trait you happen to be looking for, you know, high yield or drought tolerance or disease resistance has been improved. You need to make sure that's happened. And then you select the plants that you think are winners. And you have a choice. You can either give those to farmers or growers, or you can send that back around the breeding cycle, try and get another round of genetic gain in. And I guess the important point is that this works, but you don't have very much control over what's happening at the level of sort of the DNA. You can see what's happening in the field with your field trials, but what happens to the DNA, you have no idea. That's not a bad thing, but perhaps you can be more specific. And this is my favorite graph. So this is showing genetic gain in maize. Maize is a really well-bred crop. And so what the people that made this graph did is they recreated different maize varieties from the 1930s, 1940s, 1950s, all the way up to the 2000s, and they planted them out in identical field conditions, right? Same amount of fertilizer and everything. And so what you can see is the varieties from 1930 yield much, much less than the varieties from the year 2000. And that's a really good example of genetic gain, right? The environment is the same, but the genetics of these ones is much better. And that's as a result of how well they've been doing the breeding. So specifically looking at potatoes, potatoes are a little bit of an oddball crop for a couple of reasons. One is that they are tetraploid, which means they have 4 copies of their DNA. And so I'm going to show all the time DNA is these colorful bars. You know, usually it's a double helix, but this is just a representation of the DNA. So potato has 4 copies. Humans have only 2, as do many other plants, including maize. And what that means is inheritance is quite complicated. So whenever you cross 2 potatoes, what happens is all of the genes on these bits of DNA get sort of mixed up together. And it's quite random how that mixing up occurs. And it's also random which ones get passed on to the next generation. So you can see that, you know, the offspring here, shown here, they have very colorful DNA. So it's all mixed up. It's all very complicated. The other sort of unusual thing about potatoes is that they are typically clonally propagated. So what breeders will do is they'll get all of these offspring and they'll assess them, and then maybe this one here happens to be a real winner. Because it's clonally propagated, you can pass on exactly that DNA to the growers. So everyone gets the same thing, and you have a really consistent product. So what's happening in the DNA that makes all of this breeding work? And the answer is in the DNA are genes. And so genes are basically little bits of DNA that control processes in the potato, maybe flowering time or tuber size or disease resistance. And potato actually has 40,000 genes, which 40,000 protein coding genes, which is quite a lot. Humans have 25,000-ish, depending on who you ask. So these genes are all sort of sprinkled across the DNA like this. And they come in sort of slightly different forms. So if this is your potato DNA, this is also potato DNA. It's all got potato genes in it, but the genes here might be just ever so slightly different to the genes here, and so those are different alleles. And what happens is when you breed this potato is you get different combinations of alleles that are completely different to what was in the parents, and so that results in a completely different traits of the plants. This one has no flowers, this one's really bushy, this one's really small, you might throw it away. So you get these different combinations coming through, and it's really unpredictable how that's all going to work. So the only way really to know, have you got something good, is to do field trials, which are expensive, but necessary. And the point is, it does work, but is it the best way that we can be doing things? And so there are a few different shortcuts that we can take in the lab if you're suitably well equipped. And I've sort of split them into three kind of techniques that I'm calling genetic technologies here. The first one I sometimes refer to as like a traditional transgenic. If transgenics can be traditional, this would be a traditional transgenic. And so what you do for a traditional transgenic is you insert like a large piece of DNA, for example, a gene. So that's what that sort of would look like, is you put in this large piece of DNA, goes into the genome and does a thing. So it might, make the tubers larger or change the flowering time or something like that. And there is actually one example of a GMO potato on the market in the United States, developed by Simplot, called the innate potato. So they put in a gene that reduces the amount of sugar in the tubers, and you get less acrylamide, and you don't get this browning. So that's the gene that they decided to put in. A second highly topical technique is gene editing, or CRISPR. This is where you put in essentially you use some molecular scissors to go and just change the DNA that's already there. So if we wanted to remake this low sugar potato, what we could do is we could go and find the gene that controls sugar production and maybe just edit it slightly. So this is really precise and obviously works really well, but you're limited to the DNA that's already there. You can't put in something new like you can here. So good, but perhaps slightly limiting. And the third technique, the one where, the whole point of the talk and what we're going to be learning about and the kind of, I think it's really exciting, but doesn't get quite as much press, is what's called new breeding techniques. And so this is where you put in a transgene or maybe make an edit, but that transgene or that edit is only useful while you're breeding. So you use it during the breeding cycles, but then the potato or whatever the crop that you give to the growers doesn't have the transgene or the edit because it's not useful for them. It's only useful for breeding. So let's figure out why you would do that and how that works. So the transgene or the edit is used to speed up that cycle or help that cycle in some way. So that breeding cycle we had in the first stage with the crossing and the selections and all of that is what the transgene is helping with. It doesn't necessarily control a useful trait like whatever sugar in the tubers, but but it does allow you to improve your breeding practices. The varieties that you give to growers or farmers are null segregants. So we're going to talk about that on the next slide, so don't worry about that just yet. But a really nice example, and I think one that's quite easy to grasp, is this example of fast flowering fruit trees. So this is something that they work on at Plants and Food for obvious reasons. And so if I'm someone who wants to breed a new apple variety, I might want to, say, cross two apples together and then go through that breeding cycle and maybe do it a couple of times, getting seeds every generation. And now an apple tree takes something like 3 or 4 years at least to start producing flowers and fruits and seeds. And so if I'm going through that cycle 5 times, all of a sudden it's going to be 20 years before I've got a product to sell, which is not ideal. And so what you can do is you can put in a transgene that makes the fruit tree flower after, say, just 1 year. And so this is an example here of a kiwi fruit relative that they developed that has a transgene that makes it flower really early. That's the flower. And so what you can do is you can get through that breeding cycle. Instead of in 20 years, you can get through 5 cycles in 5 years. Now, of course, this plant sucks, right? This one is not going to make many kiwi fruit, so you don't want to give that to growers. And so what you do is you then take the null segregants, the ones that don't have the transgene, but do have the genetic gain. And so that's one really good example. Another one that has been used is increasing the recombination, so that's shuffling together. And then the final one that we'll talk about later, because that's what we do at Otago, is to help F1 hybrid breeding. And so I want to just talk about what a null segregant is, because I think that's slightly confusing, but hopefully with the examples I give, it will be clear. So I've just found this random family on the internet. We'll call that one, that can be William and that can be Kate. And William is a man, so he has an X chromosome and he has a Y chromosome. And so the X and Y chromosomes are sort of really easy to track because they have sort of obvious phenotypes in the offspring. So his 2 male offspring, we'll call them George and Lewis, have inherited William's Y chromosome. Whereas Charlotte, his female offspring, has inherited William's X chromosome. And they've also inherited 1 X chromosome from their mother. It's just a little bit difficult to tell which one they might have got. But what this means is that we can sort of track the inheritance, right? So we know that Charlotte has not inherited the Y chromosome from William. So she is a null segregant for William's Y chromosome. And so any gene that happens to be on William's Y chromosome, she also will not have, whereas his male offsprings will have it. And so as I said, for 6 chromosomes, the X and Y chromosomes is really obvious. William has another 22 pairs of chromosomes that he also has passed on, only 1 copy to his offspring, right? So he has 2 copies of every chromosome, but only 1 gets passed on, and that's true for all of Kate's chromosomes as well. So you're a null segregant for half of your parents' DNA, and that's how we use null segregants in new breeding techniques. And so this is exemplified here. You know, if you have this plant that happens to have, you know, a transgene on it that helps you with your breeding, and then you cross it with, say, a wild type plant that doesn't have the transgene, or maybe you self-fertilize it, half of those offspring are not going to contain that transgene. So these ones here are the null segregants, and those can be provided to the farmers, the growers. And so why would you bother doing this? And I think we sort of already covered that it allows that fast genetic gain. So that genetic gain is not dependent on the transgene. That genetic gain is inherent to all of the chromosomes. So this one here, even though it doesn't have the transgene, has all of that sweet genetic gain that you've worked so hard to breed in. Additionally, you know, for example, with the fast flowering fruit trees, that might not actually be something you want in your offspring. So this offspring here doesn't have the transgene, but it does have that fantastic highly bred chromosome that you've bred so well. And so that's what you can give to farmers. So that's just to summarize. I think that's been reasonably clear that you have this transgenic step during breeding, you get your fast genetic gain, but then what you give to growers doesn't have the transgene, but does have the genetic gain. Right. So what are we doing at Otago? And the answer is we're doing F1 hybrids. And F1 hybrid breeding is sort of it's an old technique for maize breeding and an old technique in quite a lot of crops, but not for potatoes. And so I'm just going to go and introduce how that works in the context of other crops before talking about what we're doing at Otago. So F1 hybrids are the first generation cross between two inbred parents. So here we have two maize parents that have been inbred. So they've been self-pollinated, like 5, 6, 7, 8 generations. And the F1 hybrid is the cross between two different parents. So that's shown here in the middle. And you can see it's much larger, it's got much more yield, and so it has better traits than either parent. And it's worth noting that this is only true for the first generation. If I self-fertilize this generation and try and collect seeds, all of that sort of gain is completely lost. It all segregates again. And so one of the real keys of F1 hybrid breeding and why it's so good is that inbreeding stage. So while you're inbreeding, it's really easy to fix these really beneficial traits and to track them because the plants are basically breeding true during this stage. The bad part, of course, is inbreeding depression. So even though you're purging out, getting rid of a lot of these sort of deleterious traits that happen to sort of show their face when you start inbreeding, you don't get rid of all of them. And so you get something called inbreeding depression. So you can see that the inbred parents are quite small in comparison to the F1 hybrid. But when you make that cross, you overcome all of the inbreeding depression. So you get this hybrid vigor. So you get basically a boost in this first generation. So it's well worth it to do all of this because you have all the benefits, but none of the inbreeding depression. And the final reason that this is a really good way to breed maize is that all of your offspring are genetically identical, right? The parents were genetically identical, so all of their offspring will be genetically identical. Potato growers, of course, are completely familiar with this because all potatoes are clonal, so all of your potatoes in the field are genetically the same already. And that's important, right? You need them to be the same so that they all make tubers at the same time and you can harvest them at the same time and everything is convenient. And I've shown this graph already. I told you I liked it, and I do, just to remind you that you get really fast genetic gain. And this graph here is just of general maize yield in the United States. They started doing F1 hybrid breeding in the 1930s. And then you can see that everything sort of took off after then. This is attributed to not just this, also fertilizer and agronomic practices, but at least 50% of the gains they saw here were due to genetic gains. And a big part of that was because they were doing hybrid breeding. So what does that sort of look like for potatoes? This diagram is a little bit complicated because there's so much DNA. But the take-home message is that when you've got confusing genetics, like you do with a tetraploid outcrossing potato, everything gets very colorful and mixed up, and it's difficult to track traits. On the other hand, when you're doing diploid hybrid breeding, it's less confusing. It's much more predictable. You can see the DNA is less colorful, which is just sort of a way of illustrating that it's a simpler way of breeding. And so you can sort of predict traits and stack in traits and design your genome a little bit more as you wish. So why don't we just go out there and start doing F1 hybrid breeding right now with potatoes? Well, if you look at how they do it in maize and why they do it in maize, it becomes pretty obvious. So in maize, self-pollination is really easy. So you just basically put a bag over the tassel, put it in the air, and then you get self-pollination. So you can do your inbreeding cycles really easily. The outcrossing is also taken care of because it has these tassels on the top, ears on the bottom, that's the male part. What you can do is you can cut off the male parts when you want to make F1 hybrid seed. And so all of the seed produced from this plant will be F1 hybrid because it can't self-pollinate. Potato is self-incompatible, so it can't self-pollinate, and it has small flowers. So this is, let me just see if I can get this to play. This is people driving tractors, and what they're doing is they're detasseling every 4 out of 5 rows. So these are the pollinator rows, and these are going to be the female ones. They collect the F1 hybrid seeds. And you can imagine these tractors are probably not cheap, but it's worth it, right? It's worth it to go to all of that effort to make F1 hybrid seeds because of the genetic gain. Right, and so that's where we come in at Otago. So me and Richard McKnight and Lynette Brownfield came up with a way to solve both of these potato problems at once with a new breeding technique that we call combined inbreeding and outcrossing. And so the way that works, it's a new breeding technique, right? So we start by introducing a transgene, and the transgene overcomes that self-incompatibility, makes them self-fertile, so we can do all of that inbreeding. At the same time, it's got like a fluorescent reporter in the seed, so we can spot which ones are transgenic and which ones are not. And so while we're inbreeding, we keep the transgene. When we want to make an F1 hybrid, what we do is we plant out these null segregants now. And so the null segregants are completely wild type, right? They're just ordinary old potatoes. And what that means is they're back to being self-incompatible. And so they don't self-pollinate. And if you do that twice, you get 2 plants that don't self-pollinate. And so if you just plant those out together in the field and allow them to cross-pollinate, all of the seed that you produce will be F1 hybrid seed. So it's a really fantastic solution with a new breeding technique for helping F1 hybrid breeding. And so this is just a nice wee picture from the lab. So we've done this. We made some true potato seed from our chow plants. So every good scientific discovery needs an acronym. Our acronym is chow. And so you can see the transgenic ones have fluorescent seeds. The null segregants are not fluorescent. So super, it works and looks cool, which is really what's important. And so that's just a wee summary of how the chow technology works. You have this transgenic stage during your inbreeding, but then your null segregant parents are used to make F1 hybrid seed. And so this is true potato seed, and it's all genetically identical. It could be planted in the field if someone wants to do that, or you could make seed tubers from these guys if someone wants to do that instead. That's up to growers and stuff. That's not up to me. And the final point is that lots of companies are sort of starting to do this now. Solinta is the big one. That's their whole core business. They're partnered with Bayer. Simplot is doing it. So Simplot is the big one in the United States. They keep secret what they're doing, but I know that they're doing this. HZPC, which is a big one in Belgium, and a couple of government-funded ones. So the Potato 2.0 project, it's got a cool name, so it must be good. And then a group in Shenzhen are doing really fantastic work. They're publishing in very good journals as well, but making F1 hybrid potatoes. And so just to summarize the whole talk, the goal of breeding is genetic gain. I think I've said genetic gain about 100 times. And you can use genetic technologies to enhance that, including new breeding techniques, right? Where you have a transgenic step, but null segregants is what's planted in farms. F1 hybrid breeding is an exciting way, a new way of improving genetic gain in potatoes. And at Otago, we're working on a new breeding technique for F1 hybrid breeding. Cool, so I'll say thank you very much, and happy to take questions. I'll stop sharing as well.
Has anyone got any questions? You should be able to just unmute yourself.
Yeah, Rowan, Ian Kirkwood here. We met and you went through this with us when we were down in Otago. I just had a question relating to the number of parent diploid species that are available for breeding. I know you've been working with Solanum furrica crosses, which are diploid, but in terms of the availability of diploid varieties for breeding, how readily available are they?
That's a good question. I don't fully know. I know that lots of the other groups have gone to a lot of effort to find amenable diploid varieties, sometimes even going into sort of wild relatives, going outside of tuberosum. The other option is to take a tetraploid variety, and you can, if you're clever, you can halve the number of chromosomes, make it into a diploid. So you can, in some cases, go from a tetraploid that you really like, turn it into a diploid, and then use that.
But do you know what you get when you do that? Because obviously you're choosing certain genes and not others.
You do not know what you get when you do that.
Yeah. So those other companies that are doing F1 hybrids, are they working with diploids as well, not tetraploids?
No one's doing it with tetraploids. I don't think anyone really wants to try making F1 hybrids with tetraploids. The genetics is too complicated. You lose a lot of that benefit.
So Solenta's been working on this, as you say, for some time. So they must have a resource of diploid varieties that they're crossing. Is that correct?
Yeah. So I don't know specifically what they've done, although they did start with a wild relative, Solanum chacoense, chacoense, and the process really is you make this cross. You start with chacoense, which is probably not a very good potato at all, and over time you introduce more and better genetics, and so it just takes a long time to develop your inbred lines. And I mean, if you look at what they did in maize, it took them a reasonable amount of time to develop good enough inbred lines that they could actually make F1 hybrid seeds. The first inbred lines that they were making were rubbish. It just takes time. Nowadays, the inbred lines that they have for maize completely outperform any F1 hybrid or any variety from back there. So it's just a matter of time and getting through the breeding cycles to improve your inbred lines.
Thank you.
Yeah, no worries.
We've just got a chat message. Can you read the chat, Rowan? Do you want?
Yeah, I'm just loading it up now. Okay, so the question is, what are the traits that New Zealand potato growers are keen to have prioritized for improvement? And how can Chow help realize these? What would you try to target first? This is a tough question when you're in giving a presentation to growers, because they will quite quickly point out that I've got it wrong. So I think disease resistance is the big one. So potatoes historically, I'm getting nods, so that's promising. Potatoes historically have had sort of lots of issues with disease because it's clonally propagated. They can build up disease susceptibility. And so introducing resistance to certain problematic diseases would be the main one. And then what you can do with Chow technology or F1 hybrid breeding in general is to stack these traits. So you can have multiply resistant crops that are resistant to multiple different pathogens. And you can do this predictably. So when you're doing tetraploid breeding, you can't predict what you're going to get. And so getting resistance to 1 or 2 pathogens is reasonably, that's okay. But getting resistance to 9 or 10 becomes close to impossible. Whereas with the diploid breeding, you can stack them up because you can predict how the genes are going to work.
The problem is
Okay.
that some of the disease resistances are multigenic and are across a whole range of different chromosomes even. So actually, powdery scab is being our number one disease issue that growers would like to solve. Is going to be a difficult one even with F1 hybrid breeding.
Can't disagree that it's not a challenge. And there's going to be some battles that you might need multiple different ways of targeting. Maybe some breeding obviously contributes to that. Fungicides, pesticides, all of that sort of thing also will be part of any pathogen management solution.
Okay, I think we're going to wrap it up now. Thank you so much, Rowan. That was really an interesting presentation.
Great,
we
thanks.
will have this webinar available. It was being recorded, so we will be sending it out in our next newsletter.
Great. Right, thanks very much, everybody. See you later.
Thanks, Rowan. Thank you.
Cheers, Ian.